Good afternoon, everybody, and welcome to today's webinar session. Good morning or good evening to some of you as well. I'd like to extend a warm welcome to all of you to the second and final event in this Genome Editing Agriculture webinar series, which is a collaboration between CropLife South Africa, the USDA's Foreign Agricultural Service, as well as our local partners, Biosafety South Africa, the South African National Seed Organization, and the ARC. You will recall at the last webinar held in May, we heard all about the different policy approaches with regards to genome editing, as well as the importance of ensuring alignment and that the regulations remain fit for purpose. Well, to follow up with this ongoing conversation, today's webinar will focus on the innovation side of things. And we will specifically look at the innovation potential of genome editing to deliver solutions and benefits in agriculture. We have an excellent lineup of speakers today. So we will all be sharing their perspectives and experiences on how genome editing is impacting innovation, and not only within their countries, but also within the different research sectors. And also we'll get to learn a little bit more about what some of these genome editing applications are in the research pipeline. And last but very important, hear how technology in general is making a difference at the farm level. But before we get to the presentations, I would like to invite Doug McCallop, who is a senior advisor to the Secretary of Agriculture at the US Department of Agriculture, to deliver some opening remarks to officially kick off today's event. So it's over to you, Doug. If you could please unmute your mic. Thank you.
Thank you so much, Chantel, and good afternoon to all of you in Africa, and good morning to those of you elsewhere in the globe. I'm Doug McCallop. I serve as Senior Advisor to Secretary Vilsack at the U.S. Department of Agriculture, handling biotechnology and a broad portfolio on trade and national security. Wanted to congratulate CropLife Africa and all of our participants for being part of this discussion. And a special hello to Dr. Henny Grenewald and to Dr. Martin Lima. I had a chance to visit many of you in person in Pretoria before the pandemic, and I wish that we were all together in person to greet one another, to shake hands, and to meet in person. Unfortunately, the pandemic will prevent us from doing that. And as we continue to improve on numbers of population affected by COVID, hopefully in the near future, we will be able to resume in-person travel and in-person meetings. But the key to all that, of course, will be vaccine uptake. And I would just encourage everyone, as vaccines become available to you, to please get one, and to do your part in encouraging the community and those in your areas to seek a vaccine when it is available to them. As we talk about life sciences, I find it very important to just sort of note for a moment the emergence of messenger RNA that many of the vaccines developed and under development during this pandemic have for the first time utilized messenger RNA as the way to tackle an unforeseen and very large problem in terms of the issues that the pandemic provided. So those tools have, I think, been shown to be a very powerful means of overcoming the challenges of the pandemic. But they only work if governments are prepared to review them scientifically and to provide approvals in a timely fashion. And I think that's a very timely topic as we delve into the issues as part of this conference here this afternoon and this morning, is the fact that as we face one of the largest challenges, not only of life sciences, but earth sciences, and that being climate change, the role that genetics and specifically genome editing will play in that issue. The Biden administration here in the U.S. has put forward that climate change will be a top priority for all federal agencies, and USDA and many of its federal partners are working very diligently on turning every solution possible toward reducing greenhouse gas emissions, and focusing on mitigation efforts to go along with climate change. Biotechnology will play a major role in this process. As we look at the current methane numbers, as we look at the current carbon dioxide numbers, we know that there are tremendous opportunities to store additional carbon in the root zones of plants. If those plants are bred and genetically changed in a way that speeds up that process or increases the capacity of the root zone to store carbon. Similarly, issues of adaptation to climate can be addressed through genome editing as well. These include issues such as drought tolerance, heat tolerance, additional plant vigor to help plants deal with changing climate conditions out there. Beyond that, food waste has proven to be one of the largest contributors toward climate change. And we know that many parts of the world that this loss in processing and the production of food provides absolute environmental impacts and certainly climate impacts as well. So the future of crops potentially having a longer shelf life, being able to be transported further distances and with greater quality, hold tremendous promise for our meeting climate change objectives in the future. But all of these solutions, if they come through genome editing, only work if governments are prepared to scientifically review them, to provide approvals, to move those into the reality of what the promise is that they can hold. Recently, the US government amended its plant regulations for biotechnology to appropriately recognize plant breeding innovation and to provide a clear pathway for products that mimicked things that could be done through conventional breeding. So many of the future promises of what genome editing can provide in the US will have a much more direct pathway toward commercialization and toward realizing some of the opportunities that they can provide in terms of sustainability, humanitarian improvements, and even quality of those crops and products. So I am very happy to be part of kicking off your meeting this afternoon or this morning, depending upon where you are, because there is so much potential, there is so much promise. It is important that we stay in close collaboration and close conversation, and that to the degree we can, that our regulatory systems be compatible or certainly able to work seamlessly across national boundaries. Beyond plants, we will also in the US be looking at the potential for animal biotechnology and what genome editing in animals could mean in terms of our regulatory approach in the US. So with that, congratulations on your meeting. This is a very, very important topic. It's very, very timely. And the future of life sciences and the future of our approach worldwide to meeting many of the most demanding challenges is connected to our success in helping to advance and empower and enable genome editing. So with that, Chantal, I'll turn it back over to you and to Dr. Grunewald to begin the next portion of the agenda. Thank you so much.
Thank you very much, Doug, for those introductions and insightful comments, especially highlighting the important role that technology has in addressing some of those climate change challenges, and also the importance for regulators in providing that enabling regulatory pathway. I think this has definitely helped set the tone for today's discussions. So moving on to the main event, our moderator for today is Dr. Henny Groenewald. Henny is the Executive Manager of Biosafety South Africa. And so I will now hand over to you, Henny, to take us forward with our discussions.
Thank you very much, Chantal. It's my great pleasure to talk to you from a cold and wet Cape Town, and I'm looking forward to a very productive meeting. As Chantal said, I manage the biosafety platform in South Africa, and our mandate is to support sustainable innovation in the bioeconomy. So this today's topic is very dear to my heart. So as Chantal also said in the initial introduction, is that this is the second of a two-part series where we looked at the policies and the regulatory opportunities in the first one, and here we will focus more on the innovation side of it. So I'm just going to have a quick run through of the meeting housekeeping rules. We're going to basically have 5 speakers that will talk right after each other over the first hour of the webinar, and then we will have all the questions and the discussion right at the end. So you must please use the chat option within the Zoom meeting to submit your questions, as we will not be able to take personal questions. Just a small note on that, you will only see your own question, but the panel will be able to see all questions, so don't worry if you only see one. So talking about the speakers quickly, please just keep an eye on the timing. I will only give you a 1-minute warning to please wrap up if I need to do that. And then to the audience, I think to me the most important part is, to emphasize that we must please use this as an opportunity, and not only submit your questions, but any other opinions or statements that you would like to make. We will be sure to gather all of this information and make it available to everybody after the meeting, and use it in any discussions where this might be viable. Finally, I must also just say that we will run an opinion poll as we progress through the meeting. So after each of the presenters, there will be a 1-minute small little break where we will ask you an opinion question, which you will then just select from the options on the screen. It's very easy. Please do that as soon as possible, that we can continue, because as soon as we continue with the rest of the talk, you will not necessarily see the questions anymore. And with that, then, it's my great pleasure to introduce our first speaker. So that is Martin Lima from Argentina. So Martin is currently an adjunct professor in biotechnology at the National University of Quilmes in Argentina. But most people here would probably know him from the 15 years that he spent in policy making and regulation in Argentina, and where he was the chair of the National Biosafety Commission. And then where one of the great things that they did was to establish the first ad hoc regulatory system for genome editing in agriculture in the world, and lots of people have followed the example since. So Martin, with any further ado, please present the talk of Martin's talk will be the Argentinian experience in genome editing applied to agriculture. Thanks.
Thank you, Henny, for your very kind introduction. I would like to thank you and Chantel in the name of CropLife and Doug in the name of USDA for this opportunity of sharing with this audience our preliminary or pioneer experience in the regulation of genetically edited organisms in Argentina. So since the time is scarce, let me just jump to the presentation. Well, to start, in my perception, it was the regulation, an adequate regulation, what created an opportunity for genome editing applied to agriculture in Argentina. Argentina has a strong biotechnology regulatory framework. As South Africa, we were both pioneers in this field, and we had the first commercial of a GM crop in 2006. The Argentine Commission is recognized by FAO, I mean the United Nations Organization for Agriculture and Food, as a center of reference for biosafety in biotechnology. And we have a real experience in having field trials and many approvals of genetically modified organisms that are used in agriculture. So in this aspect, we are quite comparable to South Africa. Then in the year 2012, we began a debate on how to regulate genome editing in agriculture. It took 3 years. 2015, we issued the regulation, and then we started to make assessment of particular cases. By today, our regulatory commission has analyzed over 25 cases and several other countries in Latin America have enacted similar regulations. Our regulatory criteria, was created on top of existing regulation. So as many other countries, we have a regulation for new conventional varieties and a regulation for genetically modified organisms. In our consideration, the genome edited organism, depending on how they are produced, they could be GMOs or non-GMOs. But if they are non-GMOs, they are completely equivalent to a mutant that could be obtained by other means like mutagenesis, chemical, radiological, or spontaneous mutagenesis. Therefore, our innovation was to create a sorting mechanism to decide if a product from genome editing has to be regulated as a GMO or as a new conventional variety. And this mechanism is based on the Cartagena Protocol definition of living modified organism that we also use in Argentina as a definition for GMO. And therefore, we hope that these assessments could be transportable to other countries also using the same definition. Fortunately, the amount of information that we need can be put together by the developers very easily. And once we receive the dossier, we can make an assessment and decision in 60 days. Having a system like this that can provide an answer so quickly and even perhaps at the same level of importance, we can give an answer at a preliminary stage when the product is still under development. This has attracted a lot of investment in research and development, not only from the public sector, but also from the private sector. So in here, you can see some examples. Researchers from the National Argentine Institute for Agricultural Research developing potatoes modified by genome editing, a startup company created in Argentina just to develop genetically edited crops. This company wouldn't exist if we didn't have this regulation in Argentina and in our neighbor countries. A seed company that is all, you may know this name because it's also active. It's an Argentine seed company, but also active in South Africa. This company didn't have biotechnology of its own. They just multiplied seeds and licensed seed biotechnology from other companies. Now they have created or installed a genome editing lab to develop their own genome edited varieties, and all of this because of the pioneer regulation that we have in Argentina and now Brazil and Paraguay. And this is not only regarding affecting crops, also our national researchers and private companies are working now on the genome editing of animals for agricultural purposes. This is an analysis of the case laws of different applications that were presented to the Biosafety Commission. As you can see, there is a lot of diversity here. Most of the products are genome edited, but there are some others, so-called new breeding techniques, that are also covered by this new regulation. And very important in here, you can see that there are many different traits. So it's not just herbicide tolerance or very basic agricultural traits. We also have health benefits for the consumer. We have increased sustainability, even improved vaccine performance or increased microbial fertilization. So the diversity of traits is higher compared to what other technologies are currently offering. And it's not only in crops, it's also happening in animals and microorganisms. And this, it's important to highlight, is not a statistics from papers or patents from initial development. These are statistics from applications to the regulatory body. So this is for real, this is for business. And you can see here also at the bottom left, the distribution of the profile for the applicants, and most of the applicants are local biotech or seed companies and public research institutions. So this has, you can see here that this regulation, since it is achievable, it can be managed by middle income companies or public research. They are actually trying to push products to the market through the regulatory system. It was not, regulation was the beginning, but it's not the only aspect to be covered. We have also engaged in public communications with the Argentine public, so they can understand what is genome editing. They can know how we, in advance, how we plan to regulate, and they can have an opinion on this. So for years, in parallel to making regulatory decisions, we have been holding webinars and seminars with journalists and with the general public, and taking their feedback into our process. That's to the inside of the country. Then to the outside, Argentina also has tried to reach out other countries and explain about our pioneer approach, and try to convince them that this is a reasonable approach. For instance, here you see a statement that was presented to the World Trade Organization. The communication was proposed by Argentina, but many other countries followed or joined. And this statement is trying to anticipate and avoid trade barriers derived from countries having asymmetric approaches to regulate these technologies. We are pledging for harmonization. Yeah, I mean, if you want to see it in detail, these declarations are online. This is another declaration, a joint declaration from the ministers of agriculture in the southern corner of South America, with a similar intention in support of this technology of genome editing being used for agriculture. And when Argentina had the opportunity of being the host of the G20, there was a side event, the Agricultural Chief Scientist meeting. And in this occasion, we also, Argentina convinced the other G20 member representatives to issue a statement in support of the future use of genome editing because of its high promise as regards to obtaining new traits for different purposes in different agricultural chains in a safe and simple way. Just to finish, although this in our region we began in Argentina it should be 2015. Then 5 or 6 years later, now other countries in our region have very similar approaches. That you can see them in blue in the map, and in these other countries, the governments are regulating genome editing in the same way, and they are seeing the same results. I mean, foreign and local investment in developing genome editing projects, and that are presented to regulators, and the regulators give a preliminary response if they will be considered GMOs or not, and these feedbacks into the support to these projects. And with this, I finish, and of course, I remain available for your questions. Thank you very much.
Martin, can you just stop sharing your screen, please? Hi. I think we will quickly do the first opinion poll question. As I said, we will ask questions at the end. We'll have the discussion at the end. So please also a reminder to use the chat option. The opinion question, please. Okay, I think we can continue. So the next speaker will be Dr. Lukeshni Chetty, who is the General Manager of the South African National Seed Organization, or SANSOR, like we all know them. And SANSOR is one of the leading seed trade associations in Africa. As General Manager, she represents the South African seed industry's interests nationally and internationally in all seed trade and policy-related matters. Before joining SANSOR, Lukeshni was the Deputy Director of GMO Monitoring and Research at the South African National Biodiversity Institute, of SANBI, where she was responsible for the GMO monitoring program. And I think this is one of the places where I met Lukeshni for the first time. So Lukeshni, we're looking forward to your presentation with the title Genome Editing in the Seed Industry. Thank you so much.
Thank you, Henny. I'm going to share my screen. Okay, Henny, is it sharing correctly?
Perfect.
Okay, thank you. So good day, everyone. I'm going to talk today about gene editing in the seed industry. And I'm going to start with the importance of plant breeding. Plant breeders have always responded to the demands for either increased crop yields, more nutritious varieties, crops that are either disease or pest resistant. And lately, innovations have simply allowed breeders to breed plants in less time with more precision. The efficiency of plant breeding has always been driven by sound scientific principles that date further back to Mendelian laws. I have a picture here of a modern eggplant, which is a product of plant breeding, without which we would have to eat this little green wild eggplant. This is a journey in time in the history of plant breeding. It starts with the domestication of wheat, moves through hybrid breeding into GMOs and marker-assisted selection. Plant breeding has evolved over the years. For the last 10,000 years, these techniques have evolved to what we're going to talk about today. What does this mean for the seed industry? The seed industry is about trade and the movement of seed, and a particular variety could be bred in Europe, produced in South Africa, taken back to Europe for seed processing. From Europe, it could go to the US for packaging, and from there, back to South Africa to its final market, which could be several countries. I think it's important to note that very rarely do we breed, produce, and sell in one country. The global seed trade is quite, and you use many countries for different things. And that's just due to climate favorabilities. But how do we ensure that we can trade efficiently? And the gentleman from the USDA really alluded to this in his introduction on harmonization. And harmonization is of utmost importance for seed trade. It facilitates trade and seed movement and across the seed industry, there are numerous organizations that ensure that we have harmonization in the various topics, such as OECD. So we have an OECD seed scheme, or we have the International Seed Testing Association, which ensures that if you're an ESTA lab, you meet certain criteria, and those criteria are acceptable. One such organization is the International Seed Federation, which is the global representative body for the seed industry. Different national regulations will require that we have different pre-market assessments, which will result in quite burdensome requirements for seed companies. Without sound scientific policies, we will impede innovation in crop breeding, limit farmer access to better varieties, decrease the availability of improved nutritious produce to consumers, negatively affect research collaborations, and reduce the diversity of genetic resources. I want to put forward the position of the International Seed Federation, ISF. SANSO is a member of ISF. We are the local seed association representing South African seed companies, and therefore we are a member of ISF, and seed associations and seed companies around the world use ISF as its global voice. This position is available on the ISF website, or you can contact SANSO for it. So very briefly, I'm going to just take you through some points. I think firstly, what ISF is saying, there should not be a differential regulation if the products are similar or indistinguishable. Adoption of sound, scientifically based and consistent policies, regulation using scientific principles. They are saying inappropriate regulation and associated costs will impede the utilization of innovative breeding methods. Harmonized policies are essential, and ISF rejects disproportionate regulatory progress. We're also saying through ISF and SANSO that there should not be regulatory oversight where there's no novel combination of genetic material, where there is it is sexually compatible species, or if the genetic variation is as a result of spontaneous or induced mutagenesis. In 2017, March 2017, an ASF document called The Regulatory Implications of New Breeding Techniques was published. And this document very clearly and very nicely outlines what should be regulated. Unfortunately, in South Africa, we still do not have regulations, and we really just hope that the department falls on this document and gives us regulations soon, as there are a number of products that can be of benefit to farmers and seed companies. What we have learned from the past, and the past being GMOs, we have learned that there's been this patchwork of regulations throughout the world, and this patchwork of regulations have resulted in asynchronous decision-making for GMOs globally. I remember when we had a drought recently, we were going to have a shortage of maize, and it was important that we had synchronous approvals, and we didn't have synchronous approvals. And we had to really get these traits approved by government so that we wouldn't have food shortages. So the implications of asynchronous decision-making can be really very dire for a country. What it results is in an environment where only large companies will have the financial capability to do these costs and regulations. You'll have very few crop traits coming out, and breeding innovations will not be available to the academic community or national research. A second very important lesson we learned is communication, or rather, how not to communicate. These are some of the images that were taken out of the GMO era of frankenfoods and scary pepper images, which frightened people unnecessarily on a product that has been so beneficial to farmers and consumers alike. One of the things that I was asked to talk about was communication when it comes to genome editing and plant breeding innovation, and what we tried to learn from the GMO communications and what went wrong there. And we decided that before we began to communicate, it would be important to understand the perceptions of the society we are in. So we collaborated with Biosafety South Africa, and we embarked on quite a large study to understand these perceptions. The first was a qualitative study and then a quantitative study. So basically, we wanted to know what they knew and then how much they knew. So in the qualitative study, we had 3 groups: millennial moms, yes, millennial moms are a thing, general consumers, and health-conscious consumers. There were 8 people per group. And then we did a quantitative study with 314 people, with 3 provinces, and the representation was according to the South African demographics. Some of the unexpected responses we received, you could say unexpected, I'm being polite, but it was quite bizarre, was plant breeding lowers nutritional content. I trust researchers, but not scientists. Somehow the word researchers offered more comfort than scientists. Plant breeding equals processed food. But in general, people were afraid of pesticides, bleaching, growth hormones, and UV lights. This is just I'm just touching on the results of the study. Results will be published next year, so you'll see that in more detail. Here on the familiarity of plant breeding, 89% of individuals were not familiar with plant breeding. In fact, some of the responses on plant breeding was plant breeding uses sunlight to help plants grow quicker, not to be mistaken with photosynthesis. Another comment on seed was, where does seed come from? It comes from a special machine, or it comes from compost. So you could see really people were very concerned about plant breeding. They didn't know what it was. They didn't know why you wanted to breed with plants. More importantly, that concerned them. Nonetheless, we pursued with this. Then we explained to them what does new breeding techniques mean. And once we explained that to them, and then we said, what if we use new breeding techniques to reduce pesticides or improve the yield of crops or extend the shelf life? Then we saw stronger positivity. Once they understood what it was and they understood that there could be benefits, you could see higher percentages. So I think in summation, I'd like to say that communication is obviously really, really important. It's important to know who you're communicating to, and it's important to understand initially what they understand so that you can communicate effectively. Thank you for this opportunity to present.
Thank you so much, Lokeshni. Oh, we're back.
Sorry.
Thank you. We can move on to the second poll question, please. And then while you answer the question, I will introduce you to our third speaker, is Dr. Glendon Askov of Stark Airs. So Glendon is the research director at Stark Airs, which is a born and bred South African seed company, which is almost 150 years old. And it's the leading vegetable seed supplier in Africa. Glendon oversees their breeding activities and their whole research and development program, including their DNA and pathology labs. Glendon will present a talk entitled The Importance of Gene Editing as a Tool in the Vegetable Research and Development. Thank you, Glendon.
Let me unmute myself first. Thanks, Henny. Good afternoon, everyone. Real pleasure to be part of this panel this afternoon. And as Henny said, the title of my talk is The Importance of Gene Editing as a Tool in Vegetable R&D. So if there are any people who are not familiar with Stark Ayres, we've been around 144 years and initially a seed trading company, buying seeds, selling seeds. 1922, Charles Stark merged with Chase Ayres and formed Stark Ayres, the name that we know today. Around about the mid-80s, late 80s, that's when Stark Ayres started its own breeding programs. We started employing plant breeders and technical personnel and doing breeding activities within South Africa for local farmers. And we've added to our portfolio over the years. And this is an example of some of the products that we now sell in South Africa. And we're, as he said, we're South African owned, we're a family company. We sell into Africa also. We have subsidiaries in Namibia and Zambia and Kenya. And as our products have gained traction, we are selling to over 50 countries in the world. So great to be part of such a company. The basis of my kind of short talk this afternoon is looking at gene editing as a tool. So tools help us innovate. Stark Ayres, we have multiple research stations. We have a team of plant breeders. We have research labs, farm managers, technicians breeding in our climate for our farmers. And we do already make use of various tools in order to be competitive, but also to get the most productivity for our growers. We know that if our farmers who are planting our seed do well, then we're going to do well. And so we use things like statistical and digital analysis software for our trials, various phenotyping aids to more accurately characterize our plants. We do pathology testing and screening to make sure that the varieties we're releasing are resistant to the local pathogens. And we make extensive use of DNA fingerprinting and marker-assisted selection as part of this process. And we can say with absolute certainty that these tools have made a significant difference in our breeding processes. We're more accurate, our breeders are more efficient, we can do things quicker, and the result is better and objective data-driven decisions when we're doing our breeding process and releasing varieties. At this point, Stark Ayres does not procure or test or develop or even research any GMOs for a few reasons. Firstly, the definition of GMO as it exists at the moment includes gene editing, and the cost of developing a gene-edited variety and then having to go through the process of releasing it is time-consuming and very expensive. Also, being in the retail space, we're aware of the stigma, as Lukeshni said, from an uninformed public and the risk of reputational damage that may result if we don't communicate these things properly and the perception in the public remains skeptical or negative. So if we have a look at the hybrid breeding process, this is what our breeders do day in and day out. They are developing populations for specific breeding objectives. They are making selections. They're looking at thousands and thousands of individual plants in their nurseries and selecting those that meet their criteria. They're self-pollinating these down for several generations until they become inbred lines. They then are test crossing between different lines to produce hybrids which go into trials. And once those have gone through several years of trials over multiple locations, we hope to release a variety and for it to be adopted into the marketplace where we can produce seed and then sell it on to farmers. And at every single one of these stages, we are making use of multiple tools to help us do things better. And so I guess for me, the big take home from my talk is better tools equals better food. And that really is true. So if we look a little bit more broadly beyond the tools that Stark Ayres are using, what are other companies using? Research organizations, universities all over the world, they're using tools like tissue culture. to help clonally propagate advanced or superior material. They're using them for double haploid production, for virus elimination, for embryo rescue, for speed breeding. They're using DNA technology, fingerprinting, marker-assisted selection. They're doing genome sequencing and coupling that with bioinformatics to develop their markers better. They are skim sequencing. They are doing genomic selection as an improvement to make gains quicker in their breeding processes. Mutagenesis, another tool that is used by a number of companies, and can be chemically induced mutagenesis, or it could be irradiation with gamma rays or with X-rays. There are recombinant tools, the classical GMO, as many of us know it, where foreign DNA or foreign genes are inserted into the plant. They recombine within the chromosomes, and this foreign bit of DNA is inserted and is active. There are gene editing techniques, which is why we're all here today, I guess. And these are a lot more precise, a lot more accurate, a lot more efficient, and have the possibility of not introducing any foreign DNA in the final product, which is a massive improvement over the recombinant ones. There are also things like digital phenotyping, using advanced sensors and cameras to capture images and assess not just the shape and the size and the color, but also looking at UV and infrared and near-infrared spectra to assess photosynthetic ability and all kinds of things related to digital processing. And lastly, probably one has been used the longest, is making use of genes from wild species. This is a wild tomato, if you didn't recognize it, and many of the advanced traits and the resistance traits have come from wild species, where we're incorporating DNA from these ancestors to help us with our breeding kind of process. So some key questions that we as a company are asking: have tools accelerated plant breeding, plant production, and have they improved food security? And we know that they have, it's without a doubt. Should we use all available tools in our breeding process? And I believe we should, provided that the end products are safe. And so if the legislation does change, or the definition of GMO in the current regulations change, so that genome editing is considered non-GMO and is subject to different regulations, then as a company, Stark Ayres would be very keen to head down this road. And just a quick glance at the literature, as Martin says, there are yield-related improvements, plant architecture traits that could be edited into plants, disease resistance, improved nutritional components, shelf life increases, etc. And I guess the last point that I want to make, this is my new favorite drink at the moment. It's a chai tea latte, and they're just delicious. If you haven't had one, I highly recommend you get one. They're really good. And a few weeks ago, I turned over the back of the sachet and had a look at the ingredients. And I've written them out here for you. I don't expect you to read them, but I'm sure you've seen similar before. Lots of not so natural looking components here in this product that make it taste great, but probably aren't the healthiest for you. And what caught my eye was this fact: there is no mention or regulation on the method or the process of manufacture of any of these components. And I checked in with a colleague who is in the food industry. He's in a company which supplies these kinds of components to many companies across the globe. And he said to me this morning that there's no regulation on the production or the method of processing. Only the end product has to meet quality criteria, has to be safe, contamination-free. And so in the food industry, this last red block over here, foodstuffs, beverages, they list the ingredients, not the development or manufacturing process. That's not important, provided the end product is safe for use. And our concern as a company is that our regulatory authorities have taken a method-based approach, where they look at the developmental method rather than the end product. And with all due respect to the EC and the AC, who have done extensive work, it's resulted in a definition where we have strict regulation that we feel stifles innovation in this area. And so unlike the food we consume, the vegetables we want to produce are going to be subject to different regulations, method-based approach rather than an end product-based approach. So those are our thoughts. And with that, thank you very much. Henny?
Thank you so much, Glendon. I want to quickly use the opportunity with some of the comments that Glendon made right there at the end. If you have any questions, remember, please, the chat box. Drop your questions in there for each individual speaker, or like I said earlier, also any comments that you would like us to highlight during the webinar. And then before we go on to the next one, just a quick opportunity to open the opinion poll for question number 3, please. Just give it a few seconds. There you are. Okay, that little bit of silence should have given you enough time to read the questions. So while you're busy finalizing that, I'm going to go on to introduce our fourth and penultimate speaker. It's Dr. Dirk Swanevelder from South Africa's Agricultural Research Council. So Dirk is a senior researcher in genomics at the ARC, at the biotechnology platform specifically, where he's involved in the implementation of new technologies within the unit. His work focuses on various topics, and one of these is genome editing, specifically looking at CRISPR-Cas type systems. And one of the places in which they apply it specifically is the ARC's sunflower breeding program. So Dirk is going to talk a little bit about genome editing in agriculture, the promises and the practical problems. I like this type of title because in the end of the day, something is only going to work if we can actually implement it. So Dirk, I'm looking forward to what you have to say. Thank you.
Thanks, Henny. Good afternoon, everybody, and thank you for joining. Thank you for the opportunity to talk today about this. Let me just quickly share my screen. Yes, so I'm going to talk about some of the promises and practices, practical problems in genome engineering or gene editing, and it actually fits very nicely with the previous talks that we've heard up to now. I'm going to start with crops since I'm an animal geneticist, but I'm also going to highlight some changes or some genome editing or gene editing events in livestock. Here is just a summary view of current publications in crops important to Southern Africa. I tried to capture some of the genome traits that were targeted. Glendon, I've included tomatoes for you. And just to highlight some of the areas that are researched at the moment is often related to traits important to the consumer, to storage, like Doug has indicated earlier, a storage or better production for a trait for the end user. But also there's sometimes for breeding objectives, specific traits addressed. For example, in green, induction of haploids. Diseases are also covered in red. And then interesting tomato domestication, where they used a wild tomato that Glendon showed earlier, and they've changed some genes or edited some genes and domesticated about 60% and got more flavor and fragrances out of that tomato. Other crops, just to broaden the horizon of papers that are actually out there, also focuses on these different traits important to your end consumer. The mushroom anti-browning phenotype was a FDA CRISPR release where they actually release a product by the FDA in the US that is not browning of mushrooms. So you have a consumable food product that the end user can use longer, less wastage. Some of the traits are across species applicable. You can, for example, herbicide resistance is often used to set up the different experiments where a single point mutation can result in herbicide tolerance. It's been done multiple times in rice. It's been done in a number of other crops, and this could be potential for some of the indigenous crop species in South Africa for that type of trait. If you want to produce herbicide resistant indigenous crops. Moving on to livestock, the livestock groups have focused a lot of animal welfare. Some of the first work were done on dehorning. There's a dehorning or poult trait. Poult is no horns, a natural mutation in some of the beef cattle. And basically the idea was to remove the horns from cattle for their own safety, but also for handlers. And breeding it into milk cattle or dairy cattle would have been a long and time-consuming effect. They talk about 50 years plus, but they were able to do this with gene editing with no loss in milk production. Another animal welfare area is where specific genders are preferred in a production system. You might want to have a high number of beef bulls in your beef cattle, because they just bigger carcasses, more weight, and they grow faster. The one that sort of made me cringe was genetic castration that they use for pigs, where they actually genetically castrated the boars, and they use those as then as surrogates to re-implant from elite boars sperm-producing cells, and then you have an elite boar sperm-producing surrogate. You can also just produce female piglets now in some of these experiments. Of course, productivity is also important, just as in the crops. And here, the myostatin gene is a natural occurring mutation in mammals. Even humans can have it, forming double-layer muscle tissue. And they've actually introduced this into merino, and they got higher carcass weight without any loss in wool quality. Same with cashmere goats, as well as with pigs. Pigs, you can now get double the bacon with the mutation. Disease resistance in animals is a little different than in plants, especially diseases where you do not have treatments, or you have to basically cull your whole population or production system and restock from the get-go. These type of genome engineering opportunities are really interesting to the community. For South Africa, African swine fever is an interesting case where they've found warthog to be asymptomatic or to be resistant to this fever. And they've found that if you introduce these variations that you found in warthog into domesticated pig, you can actually introduce resistance. Interesting enough, you can even get now corona-resistant pigs. I had to put it in here. By the way, this was developed in China. But you can see there are a lot of interest in the animal side as well to produce better quality and more humane ways of producing animals. I've included biomedical for two reasons. One, I think it could be an agricultural opportunity in the future if you have a small specialized farming system where you can produce, for example, organs for heart transplant or transplant to humans. They've actually modified some of the genes that caused rejection in humans, in pigs, and you can now do a transplant without the negative impact of drinking medicine afterwards to prevent rejection. You can even now study human deafness in sheep. They use sheep as a model. So the benefits for genome engineering, as mentioned before, is it allows us precise way of making changes without the linkage drag. You only get what you want. You don't have to bring all the other things along that is with that gene in the chromosome as you would have with a normal cross. It's usually faster. It allows you cross-species trait movement. For example, herbicide resistance can remove meat production between animals, same genes, and it allows you to target multiple genes for systems at once. So what's the future of genome engineering in agriculture? I think we're going to start talking less about plant breeders or crop breeders, livestock breeders, but more about crop and livestock engineers in the future. You will have a database with traits, the associated targets that you need to change. You'll go to your elite crop or your elite line, select those genes and those traits that you want to change that, and you will engineer your final product instead of going through a long breeding process. In the end, you will verify this probably with whole genome sequencing and digital phenomics, like Glenn have mentioned. And then this will go into a standard process line. And as Martin has mentioned earlier, I think these are, if there's an enabling environment, this will move much further forward and faster forward. Now, this is also the view of the economist. Here is a projected growth for the next 10 years in crop biotechnology, specifically in the seed market. And the area that the area that is predicted to grow very substantially in the future is gene editing. So this clearly shows that this is a really important area to invest and to be involved in. So the question is South Africa ready for genome engineering or gene editing revolution? And I want to actually talk about my own experience here. The last 3 companies that approached me around this topic were mostly interested in CRISPR-Cas or CRISPR-type work. 2 of these were startup companies, which were very, very supportive of this small company driver mentality that's been presented, or evidence that we are seeing. And one of these were actually in livestock. And the discussions always start with these fantastic benefits of the technologies. And then we start talking about risk. Initially, you talk about the technicalities. Which technical risks do we see? How can we get into your organism, first of all, or the line or the crop of interest? How efficient is the methodologies? What happens if you're busy with a system and a new technology is released? We've seen with the sunflower that it's difficult to get the final step, the rooting, done. So you must have the correct lines, but also while we're busy with a study, you can get 1 or 2 or 3 papers releasing better ways of doing what you're busy with. And then the actual showstopper, I would say, is the regulatory uncertainty. As soon as you mention this is under the GMO Act, there's a question immediately: is my project GM or non-GM? What's the time frame? What's the cost going to be? And then this association with this negative GMO tag that Glenn has talk about. By placing the new technologies under the GMO Act, you immediately imply this is genetically engineered, is GM. Just by implication, that's the view, that's the implication or association that's made. So is South Africa ready for genetic engineering revolution?
Sorry, Doug, we've got a minute left.
Thank you. I would say yes. I think we're already in the research space. The GMO Act really is supportive of getting the projects out there. However, commercially, I'm putting a big question mark here. And why I'm saying that is we need the clear guidance to what's in and what's out of the regulatory scope, for the simple reason it impacts on that first step that a small business make, and that is the project plan or the business plan that you need to present to your funders. If you don't know whether it's going to be GM or not GM, which technologies could be used that will not make it not GM, could mean thousands of rands saving or a shorter path to a product. So this is really important that we understand that we need this regulatory guidance documents to enable us to do this. It will lower uncertainty and therefore risk. And I think it will definitely stimulate the good growth in investment. And by the way, the 2 small companies actually did not proceed with work, and the larger company even stepped back and say they have to think about this. So we don't see this investment, and that really stopped the driving of the technology on the research front and therefore into projects. Thank you very much.
Thank you very much. I think we will move in directly to the 4th opinion poll question, please. just give you a few seconds to read it. Okay, then I'm going to continue just quickly by saying thank you for the questions that are rolling in. Please keep them coming. There's already interesting discussion going on there about the communication aspects, especially regulatory harmonization, which is, I think, always a hot topic to discuss. And then the whole issue about that both Dirk and Glendon touched on now is the whole debate on what's your regulatory trigger. Is it a process or a product type threshold? So yes, please keep those questions coming up. And then presenting, last but not least, our final presenter, which will always have a soft spot in my mind because it's a farmer. So Sililizwe, we are very happy to welcome you here. So Sililizwe is the managing director of an organization called Rocky Park Farming Group. And he himself is a commercial grain farmer based in the Eastern Cape near a small town called Ugi, where he plants maize, soybean, sorghum, and oats. He's also a founder and chairperson of a development non-profit organization called Ukanyo Farmers Development, where he and 35 other young agricultural graduates mentor, upskill, and develop Black commercial farmers in the Eastern Cape. He's a young farmer and development specialist who wants to see inclusive and sustainable agriculture sector realized through partnerships, new opportunities, and an environment where farmers are allowed to do what they do best, and that is farming. So thank you very much for that last sentence. I like it. I always also say, horses for courses. If we allow people to do what they do best, it's a blueprint for success. So Sililizwe, we are looking forward to your presentation, which you entitled Genome Editing in Agriculture from the Farmer's Eye. Thank you so much. You're still muted, sorry.
Sorry, can you hear me now, Henny?
Perfect, thank you.
Sorry, I just want to share the presentation. I hope everyone can see it. Thank you, Henny, for that. I'll just dive straight into it. Thank you for the opportunity, and I'll just obviously give a very practical opinion and perhaps mindset with regards to what everyone's been speaking about, and I've been listening very attentively, and I'm very interested, and should I say it's a learning curve for me as a farmer, which is always important, growing a business of agriculture, but also developing other farmers and bringing such technologies into that development work. My brief presentation is just from the farmer's eye view, and I'll speak predominantly of my understanding of the agricultural sector, but also cover aspects which I think are important as we go through this particular introduction of genome editing, or should I say, GMO, which has been part of the farming environment anyway for a very long time. We're sitting with an agricultural economy that's a 28.6 GDP contributor. That's absolutely massive in my opinion, and I mean grew by 15% in the fourth quarter. It gives you an indication of where and how big, or should I say, expansive the agricultural industry is, and obviously, the value of that at 74 billion, you pretty much get to understand, the value of the economy brought in by agriculture. COVID-19 pandemic, being a prime example of the importance of agriculture as an industry. An industry that I think everyone would know, specifically the guys in South Africa and people who are working with agriculture, that it's a fairly well-structured industry with main commodities, horticulture, livestock, agronomy, citrus and the like being leading commodities making up the agriculture industry of the country. Through the fact that being from a marketing perspective, a well-structured industry in its entirety. The production reality in our view, or my view as the farmer, is that we've got a massive feed crisis, should I say. When I say crisis, I'm talking about the challenge really to feed, some of the figures with regards to population globally and nationally as well. I mean, with 60 million people depending on farmers, for food, you get a quick understanding the amount of, responsibility one has as a producer. Let it be, as small as a half a hectare for your household to, as I say, 40,000 big commercial businesses or big commercial operations in the country, are actually producing food for this particular population, as well as the agribusinesses who process that food, to put it on the table for us. So I think the situation that we find ourselves in, makes it absolutely, hard to ignore technology such as GMO and genome editing, in my opinion. And it's a technology which I believe should be something that is absorbed by farmers, but also as industry, so that we actually achieve food security and maintain our food security as a country. But ultimately, there's pointers which have come from various presentations before mine, speaking about the importance of plant breeding technology, human health, sustainability, environmental aspects, which I think as a farmer are very important to you, but also, from a technology perspective regarding genome editing, it is definitely something that one needs to have, as part of the operation. It is happening already in terms of the operations that are currently happening. I think I had to understand what genome editing was as a farmer, as a basic farmer on the ground, which is the reason of the definition at the top there. But I think most importantly for this particular slide, agriculture for me, countrywide, globally, has never been a complete 100%, natural process. Research has been going on for years. I mean, Stargaze is 144 years old. You quickly get an understanding of these big, seed producing companies and seedling producing companies. They've been doing research for a very long time. So I think it's never been something that's new to us. And it's definitely not something new to us as farmers on the ground, because after all, we've got yields to meet. We've got averages to meet, and we've got obviously, investment per hectare. We need to be able to get revenue per hectare back. So I think ultimately for here, it's a situation where farming has happened. Some of the crops that are being planted in South Africa are not native in South Africa. But they've been imported, and the marketing systems, maybe the word scientist, as one of the panelists indicated, is not a really loved word. But from a researcher perspective, it's all happened that all of these crops have come in. They've been manipulated DNA-wise for us to be achieving the 8.5, 10-ton yields that we're getting as grain farmers, particularly, at least from the operation that I'm doing and the work that we're doing in the Eastern Cape. But I think the most important thing here is that this particular technology of genome editing to us as farmers is answering critical components of our production system, which includes, weeds, the preventativeness against weeds, diseases, insects, and other pests. So I think from a scientific innovation perspective, gene editing obviously, offers a better protection, as it were, with regards to harvesting from a yield perspective, while also minimizing the impact from network, or should I say, can everybody hear me? Am I still around?
Yes, it's perfect.
Hello?
I can still hear you. You hear me?
Hello? Can I just put off my camera for the sake of stability? I can hear you, Henny. Can I put off my camera just for stability?
Yes, it's perfect, because we're looking at your slides actually, so it's perfect.
Can I carry on, Henny?
You can carry on. You clear?
Thank you. I'll just go over, obviously, in terms of continuing from a genome editing perspective. Yes, I'm here. Can you hear me? Henny, can you hear me still?
Yes, we can hear you.
We can hear you. We can hear you.
Can I just put my camera off? I just want to put my camera off. Sorry. Me and see me now, Henny?
Yes, perfect. I can hear you clearly.
Excellent. I'll just carry on from here. I think it should be better with my camera off.
Yeah, it is.
Genome editing from a farmer's viewpoint, I was just busy speaking and alluding to bringing it down to more the Eastern Cape, predominantly. It's a situation where, for me, in my understanding, Eastern Cape is one of the poorest provinces. And the reason why I'm talking like this is because, as I had alluded to in the top, we are a province that has got high opportunity, and we are a province that is facing high unemployment. So link those two things together. You have to look at ways and how to change that particular picture. So in my understanding and my view is that we have got food production to answer, and we've got agricultural potential to unlock. That automatically means, in my view, the importance of a technology like genome editing being part of the realistic, picture turnaround, as it were, with regards to that particular work. Some of the work that I do pertaining to development work is obviously commercial production of maize in rural areas. So this automatically means the type of crops that we're producing in the rural areas is of commercial standard. And yes, we are using GMO plants or GMO seeds in that particular way. So we're in a position where currently Eastern Cape, the grain farmers on average, including my farm, we're averaging 10.5 tons a hectare of maize, 3.5 tons a hectare of soya beans. That's the type of tonnage we're achieving. Why? Because we are using obviously genetically modified seeds in terms of our planting with regards to the work that we do. So in a nutshell, it just again points to the importance of this particular research, of this particular technology. And Rocky Park Farming Group, the farming operation that I own and run, we're 1,400 hectares under dryland production. We're in a position where we've got heavy investment with regards to technology. We are a farm that is operating under a sustainable manner with a clear emphasis on achieving our targets with regards to yields per hectare. Now, the moral of the story here with regards to Rocky Park Farming Group is that we've invested in technology and we're working with technology, such as genome editing, or should I say genetically modified seeds, with regards to our planting. And some of our crops, as I had said, from a yield perspective, at 10.5 tons, when you put down fertilizer for 8.5 tons per hectare, automatically that says, with good weed control, control and resistance, pest control and resistance, we're in a position where we are able to get back that investment per hectare. But the most important thing in this particular instance is that the reason why we're able to achieve such yields is because we're using, genetically tweaked with seeds with regards to our operation as a grain farming operation in the Eastern Cape. As I close off, there are some key considerations I would say as a farmer are important as we go along with regards to enhancing food production, but also making sure that we're working within the parameters of regulation. We have to intensify the regulation around genome editing. It could potentially get out of control if it lands in the wrong hands, I suppose, with regards to people trying to take a shortcut around it. There has to be a strong emphasis on human health, ecosystem and natural habitats, and continued research is paramount, in my opinion, with regards to this type of work. Genome editing products for safety, consumption, and their long-term effects. We as farmers need to be privy to such information because our aim is to make sure that 7.9 billion people are fed year in, year out. So I think it's important that such information is simplified for us farmers to say, chaps, these are the seeds on the market. This is the type of cultivars on the market. Continuous trials around that is important aspects of ensuring that we're achieving, the type of yields that we're achieving. So I'm talking about something that's already happening, ladies and gentlemen. It's not something new with regards to using seed or certified seed that is in this regard with regards to GMO and the editing thereof. In conclusion, in my view, genome editing in agriculture is a needed reality. Times have changed. The days of hoeing for weed control and using open pollinated, seeds is something that we have to face the reality that things have changed ever since. The climatic conditions we're farming in are not the same. You know, as a dryland farmer, I depend on rainfall. You know, not to have germination and to have seeds that are not in a position of giving me that germination percentage, you know, hits me back per hectare with regards to my planting population. So all of this for me becomes important as a farmer on the ground, but also working with other farmers. Editing is already a part of our lives, as I've said. And to me, it's a necessary approach, genome editing. And can you hear me? Am I still on?
Yes.
Thanks, Henny. As I close off, sorry. To me, it's a necessary approach that can be adopted by farmers. Such a technology such as genome editing is the future. We must just simply embrace it and carry on producing food for our nations. Thank you. Thank you.
Thank you. Thanks. Everything's back on my side. Zinlizwe, you broke up a little bit right towards the end, but we got your message. So we're at the stage where we will quickly ask the last opinion question, please. And while it's there, a last reminder to please send in your questions, because straight after this, we will start discussing those questions. Okay, I think we can continue. So this is the opportunity to interact with the panelists, please. I think the first very practical question that we got is people asking about the availability of the presentations. As far as I remember, all of the presenters agreed to share their slides. So I assume that Chantal will manage this process. Chantal, when you conclude in the end, please just remember to let us know exactly how and where people will be able to access the slides. So the slides will be available to everybody. And then there's a few questions. So the last time I was only an attendee and I couldn't see all the questions. So I assume this is still the case. So I would just like to give people that have already answered some of the questions in the chat box. If everybody can't see those, I just want to give the presenters the opportunity to answer it on this platform, that we can give everybody the opportunity to hear what they are. So one of the questions that was asked there was directed at Lukeshni and asked how much of a barrier is consumer miseducation to getting approval for GE crops. Sorry, I'm going to interrupt myself quickly for this while I see this is one of the notes that I made myself as well. This GE crops now, I assume, refers to genome editing. And I think one of the things that we realized over the years with the communication, as Lukeshni referred earlier to GMOs, but now also that we're busy with genome editing specifically. And then we also have this very complex discussion on synthetic biology, for example, at the Cartagena or the CBD level. Now I forgot what the CBD stands for biological, the Convention for Biological Diversity. So the point I want to make is it's very important how we talk. And GE is a recognized acronym for genetic engineering, especially in the US. We didn't use it very much in South Africa and even in Europe. But in the US, and I would argue Martin can answer this, but in Latin America as well, GE is genetic engineering. Genetic engineering means something completely different. So long story short, there's a big drive across the globe to try and get a recognized acronym for genome editing as capital G, capital E, and then a lowercase d for genome editing, short. So with that interlude, I'm coming back to the question again for Lukeshni, asking how much of a barrier is consumer miseducation to getting approved genome edited crops and wider implementation of this technology? Do our legislators take consumer views into account? So Lukeshni, can we start off with you, please?
Kenny, thanks. I'm not aware that the regulators take this into account. I'm not sure if anybody else is. However, misinformation is bad nonetheless, and it could destroy a technology before it even becomes available. How the regulators make their decisions, I'm not sure. They are supposed to be consultative processes. The whole ASAF report was consultative as well. So I don't know. Perhaps there's a regulator here that could answer that question.
I don't have the list in front of me, so I will just quickly take the opportunity to in the first place, say in the South African system, I will focus on, but it's true of all regulatory systems around the world, is public participation is a central role of regulation in the first place at the policy development stage, which in South Africa, again, through legislation is a participative process. And then in the second place, we specifically have the opportunity when from the level of field trials, as everybody would know, and then definitely on commercial release applications, you have to do a public notification, which basically means you have to publish it in 3 national papers, and then the public have the opportunity to submit their comments as well. So that's a formal part of the regulatory process. There's another one that's a little bit hidden in the South African, it's not really hidden, but because it's not directly under the GMO Act, people often forget about it. And it all relates to labeling, for example, because you'll remember that we have this eternal discussion about labeling GMOs and non-GMOs. And again, please remember, I'm only talking about GMOs now, not about genetically engineered crops, because in South Africa, we have not finally decided or published a framework on how we're going to handle those. But for genetically modified organisms, they also have to be labeled under the Consumer Protection Act. And the big argument there, this is not a safety label, because we have safety labeling under the Department of Health, actually, since 2014, which has not been regulated because it's based on safety issues or what's the word? I forget now, but I'll come back to that. But it's information. So that's the basis of the Consumer Protection Act labeling, is to give people the opportunity. So it's not directly. So the question here is about, have the people opportunity to participate in legislation, actually in decision making? And as I explained, definitely they do have, but we also do it at the level of, giving people the choice. In the case of GMOs, for example, it should be labeled as a GMO, the product. So consumers then do have the opportunity to choose the GMO product or not. Is there anybody else? I'll just quickly a second. If anybody wants to add anything to this question about the role, we can broaden it a little bit about communication and the public's participation on the one side, but then also the acceptance issue, the social license, I think is a term. Martin, I see you putting up your hand. Thanks.
Thank you, Henry. In our experience regarding the public involvement, it was very important that there were some national developments on genome edited products. So when we were addressing the public, for instance, we have a major event featuring the Minister of Agriculture as the regulator and the Ministry of Science as a presenter of national developments. So they personally explain and engage with the public, both with an audience of 2,000 people present and online. And the reaction of the public, when you also tell them that this is not just a foreign technology that is going to be used by the farmer, but you explain that there are national developments with the faces of Argentine scientists and Argentine entrepreneurs, and that some of the traits are for instance, for the benefit of consumer, that is quite influential in the way that they address or perceive the issue. So I think that should be important also in South Africa. It's also about telling what is happening locally.
Thank you so much. Anybody else want to talk about the communication? Dirk, I see your hand, please.
Yes, so a couple of years ago, I actually presented on new plant breeding technologies to the research group, which consists of researchers, postdoc students from MSc, PhD and postdoc levels. And we actually asked, what is a GM? What would you consider personally to be a GMO? With genetic background, all of these students and postdocs have genetic background. And the arguments and the discussions afterwards were going on for months in the tea room about each person's, what would they consider personally to be GMO for them versus what the definitions say. So I think we need to keep in mind this is sometimes highly technical and how this is communicated. I like the way Martin has said they're doing it, but how it's communicated is often a technicality that makes a difference. But it was quite fun to see the students every lunchtime chatting about, is this now a GMO? And then you just walk past the tea room and you hear people arguing about what would they consider to be GMO. And I think it highlights this knowledge gap. If postgrad students have arguments or discussions about this months after a presentation, it sort of highlights, what would the general person on the street think about this? And I think we need to make sure it is not necessary as straightforward as we would think it is, because we've got the background to answer it or to discuss it. So keep it in mind, but I like the idea that Martin suggested that this comes from a government point of view. This is developed in South Africa or in Argentina, for Argentina, by Argentinians, for the greater good of Argentina. I like that. I think it's a very well thought out way to communicate the new technologies and that it's safe.
If I can just add one comment to what Dirk said, I think the important thing in the end of the day is to decide and have a clear interpretation of what your definition is. I agree 100% that obviously there can be a huge, subjective perspectives on this. But the point about regulation is actually to define something in such a way that it makes legally sense, something that you can enforce in the end of the day. But I'll leave it there now because I think the next question that I have lined up here aligns to this. And I have not had time to look through your answer, Glendon, but there was a question directed at Glendon to first say that the discussion on product versus process regulation has been with us for some long time. And then the question is, one can only expect that it would intensify as the technology develops and new products come into line. In your view, how should this be approached going forward? Would it be feasible to start looking into a dual approach to this process, regulation-wise? What would be the pros and cons to this approach? Thanks, Lennon.
Yeah, so great question. Thanks, Cloud. I think for any new products to come on the market, the regulatory space needs to be in a place where the barrier to entry is low. Otherwise, everything we're doing is under gene editing is considered a GMO, and pretty much only the big multinationals are going to play in that space. And likely the traits will be restricted to grain crops because those are the highest value and because of the highest volume. I think for us in the vegetable industry, where the product is directly consumed, you eat your tomato directly out the salad, it's not processed like with many of the grain crops. I think our awareness of the public perception is heightened around GMOs. So for us as a company, unless this is changed, we're not going to be taking the risk and the leap into a big investment where there's regulatory uncertainty. So I agree completely. A dual or a separate approach is needed for regulating genome edited products. The pros and the cons, I think the pro of having a separate pathway for releasing these things is a much simplified process. Releasing the variety, very little or zero regulation needed, as a number of the speakers have mentioned. And I think the cons well, we're going to have to educate the public on a whole other new technology. They're just not even understanding GMO yet. And we might have to educate them on gene editing and why it's safe. So pros and cons on both sides. But when you're looking at, for example, vegetable industry and companies our size and smaller, significant way to go before we even kind of look at that.
So Glenden's answer focused more on kind of the impact of what they want to have. Until we can have the discussion at some later stage, I just quickly want to make a point about this product and process. In terms of formal risk analysis or the regulatory process of risk assessment and making decisions, the whole process product discussion is actually irrelevant because the risk analysis process per definition takes both into account. I think what's confusing is that the product versus process interpretation is primarily focused on the definition that you use to determine your trigger for regulation. Because if you have a process trigger, you increase the scope of regulation, whereas with product, you limit it. Martin, I heard you make a point, so please tell us from your experience.
Yeah, thank you, Henry, and sorry for taking the floor again, but what you were saying is very relevant. We after thinking a lot about this and looking at it from different angles and trying to build a bridge with the regulation of conventional products, what we found, the GMO regulators, what we finally understood is that the conventional products also have a regulation of their own. So you may have let's say a herbicide tolerant crop through mutation or through the older ways of doing mutagenesis or through transgenesis. So in my country and in yours and in many other countries, that will decide on which regulatory system they enter. But the potential risk to the environment is the same. It's a herbicide-resistant crop. So possibly, for instance, it theoretically could transfer that to a wheat. In Argentina, when a mutant or a conventional new variety is presented to the register, to the variety register, if the trait raises a risk hypothesis, they do a risk assessment. So it's not like those products are not regulated. That's something very important to understand, especially for those that like me that knows a lot about GMO regulation and very little about conventional product regulation. So they are also regulated, but the way that they are regulated is more practical. If there is a risk hypothesis, the seed register will focus on that risk hypothesis and request information on that risk hypothesis. And if there is no significant risk hypothesis, then there is no need of making all the fuss about that. In contrast with GMOs, perhaps for the same trait, we have a lot of pre-existing, a shopping list of risk hypotheses, most of which do not apply to a particular case, but nevertheless, we ask for data about that. So the reason why one products are more difficult to take to the market compared to the other, it's not nowadays we can understand it's not related to a risk factor. It's a difference in the way that we manage the bureaucracy around them. And that's something important because the public must understand that, and people must, in general, must understand that if a product improved using genome editing is not considered a GMO, it will still be registered and possibly subjected to a risk analysis if necessary. And then you said something before about the use of genetic engineering. In Latin America, fortunately, we don't use that for regulatory purposes. Maybe they do it in the academy a lot, but not regulatory or policy making space. But in connection to that, it's also important that we avoid using a language or explaining things in a way that we suggest that we have a third category here. So we have a pre-existing framework for conventional new varieties for GMO, And now with genome editing, we can have one or the other, but there is no third thing or a third category of products. Thank you.
I think the last point that Martin made now is very important. Quickly going back, while you mentioned the terms as well, just a comment from Dean Ulofse on the comments here that he's our representative at the OECD biosafety meetings, and he said there also the consensus from the OECD meetings is that terminology is important. It must be standardized, that everybody has the same definition and understanding. And I cannot agree with him more than this. And the current discussion under the CBD on synthetic biology and what they seem to include and not include is a very good example of how this can lead to uncertainty. And then there were two questions. I must just try and find them both here again because they move from open to answered. So one of them is by Joan Foster that asks, what limitations will there be with regards to introducing traits into non-native species such as maize versus species which have wild relative or indigenous crops maybe? And genome editing versus classical GMO modification. So that's his question, but just before I give the floor opportunity to answer, there's another one from Amos Awuzu, who asked what could be the possible problems with GMO crops crossing with OPVs in the same field. So again, yeah, it's just two quick things that I want to highlight is that Almos's question here specifically is about GMOs and crossing with OPVs. And then John's question is about the potential distinction between genome editing in non-native species like the crops that come from other centers of biodiversity versus indigenous crops. Is there anybody on the South African side on the panel that wants to take on this answer?
I'll give it not quite sure I have the right answer, depending on how I read his question, but I took his question to mean, are there any risks in which is
and then you're breaking up, sorry.
Introducing.
Can you just start over, you're breaking up?
Let me. I do have a fluctuating signal here on the farm.
You sound perfect now.
I don't know if that's any better. Okay.
Yes.
So I understood John's question to be, are there any difference in the risks or limitations when we're introducing a trait into something like maize, which has no wild relatives in South Africa, versus a crop that we might plant that does have wild relatives? I hope I've understood the question correctly, John. I think my answer would be it doesn't matter too much whether you do gene editing or GMO, classical GMO. I think it depends on the nature of the trait, which would determine the risk. Would we be worried if herbicide tolerance was transferred to weeds? Absolutely, we would. Is that a problem with something like maize that has no wild relatives here? Most likely not. So with wild relatives or with crops with no wild relatives, the risk is pretty much very low. But for a crop that does have wild relatives here, we would be worried if something like that transferred to a wild relative or a weed. Would we be worried if we transferred nutritional benefits to the weeds? Probably not. So for me, it depends on the nature of the trait. Other question is kind of similar. What happens if we have cross-pollination between a genome edited or GMO crop and its OPV? Well, that seed would contain, GMO DNA, which would pass on that trait to the progeny in a normal Mendelian way. So it depends on where that seed is going to be used. But yeah, it would be transferring that trait across.
Just quickly from my side, what Glenn started to discuss here is exactly the stuff that you look at when you do environmental safety assessment for GMOs. So I think trying to keep it as short as possible, the big distinguishing thing here would be is where do you regulate it? If it's GMO and you do it's subject to the GMO Act in South Africa. And then, of course, you have to do these things as part of your environmental risk assessment. Like Martin actually said earlier, when we talk about regulated as a GMO or not regulated as a GMO, if we say some genome edited crops are not regulated as GMOs, it doesn't mean they're not regulated at all. So things like herbicide tolerance or insect tolerance and all of those kind of things are considered in a different system there, with the cultivar registration in South Africa, for example. So it's risk assessments that's done in different ways and under different pieces of legislation. But I think the crux of the question here that you ask, there's a practical side, like almost asked, can a gene flow to an OPV? And yes, it can, but it can flow from anything else. But this is what you consider while you're doing the environmental safety assessment, to actually assess if that movement has the potential of causing harm in the environment, in very short language. And we can obviously follow up this discussion later. The important point is just, so it's in the first place, there's a practicality side of it, of determining environmental or potential environmental impact. But then the legislation under which you do it and exactly how you do it, then depends on which acts or regulations it's subject to. I'm just trying to go through all of these. There's a question from Kingston Machingadze from the ARC in Potch. He say, may I ask a question, please? Michael Baru from the ARC Vegetable Medicinal Industrial Plant Campus. Oh, maybe it's somebody under Kingston's name, sorry. The question is only in the second one. It has always been the case that whenever the regulatory aspects of genetic technologies are discussed, the transatlantic effect on Africa remains on the table. As one colleague once put it, if we become like a pendulum. When the Americans speak, we swing west, and when the conservative Europeans speak, we quickly see north. What does Africa, or South Africa for that matter, has to do to have its own regulation? And this question is from Michael Beru from the ARC. So that's a good question. It seems like we're blowing according to the wind, as Michael said, and his questions say, what should we do to get our own regulation? Anybody else? I think Lukeshni, I don't want to put you in the hot seat, but you're in an organization that look at these things. Maybe if you can just give us your personal opinion on this, please, in the first place. And then the second question I would like you to add to that is just maybe also distinguish for us a little bit between the seed industry and what it means, regulation and harmonization for the seed industry versus harmonization in the grain, the commodity trade.
Okay, I might just have to ask you to repeat that second one again. But for now, just to go to Michael Beru's question, I think we have to remember, well, you're asking what does South Africa have to do to have its own regulation? I think the regulators are fully in charge and they can do what they want to do. But we have to remember that South Africa, as with any other country, is part of a global trade environment. And it's important to be a part of that global trade environment. We need access to products from other countries, and other countries want products from us. I don't really believe that we swing left or right, actually. If we look at our GMO Act, it's neither left nor right. It stands completely on its own. And in fact, what we are trying to say is at least go in one direction. There is no need to continuously reinvent the wheel because we want to feel like we want to stand out. While we feel like we want to stand out, there are companies that are being disadvantaged. Dirk alluded to that. Glendon alluded to that. And we have to be, if you want to be in the game, you've got to put on your big boy pants and play in the game. And we can't be continuously sitting on the fence. We don't know what's a GMO. Oh, it could be a genome edited product. Is it no, this has to stop, and we have to take a stance as a country. Henny, what was that question? Distinguish.
It was just the difference between seed harmonization and grain harmonization.
So seed, yes. So trade in the seed industry, those harmonized regulations refer to seed certifications, so that in Africa especially, we have the SADC harmonization system, and then we have COMESA, and that basically is to say that if you produce seed under a specific set of criteria, and if it meets these criteria, you can easily move particular varieties across a certain set of countries, which means access or trading is easier and farmers have access to these products. So a farmer in Ethiopia, for example, can have the same access to products that a farmer in South Africa has. And that's basically what harmonization means. I was recently actually asked the question about what would harmonization benefit? And really, we have these systems in place. It's just about the political will saying, yes, I agree, we can harmonize. It doesn't mean there's no rules, not at all. There are rules, we just have to adhere to it. And you have a harmonized process. Political will, in my opinion, for that. I hope I've answered any.
It's perfect. Thank you so much. I like it. I like that take-home message that you threw in there right at the end, is that it's political will. It's important. We need, to make the decision, one way or the other. I think there's only 8 minutes left on my clock, so unfortunately I will have to stop. There's still a few questions. Daisy Kapalbo of Imbrapa also asked a question here about the likelihood of harmonization across the world, how likely that would be. This is the type of discussion that we discussed more in the first one. I think this just reminds me, and this is why I emphasize this question as well, is that it's a lot of the talk that we did. Remember, we're talking about innovation here. I see a lot of the people that participated are research and development, academic researchers, people involved in innovation system. But still, the whole story about regulation is central to this. And this is unfortunately what I see in South Africa as well, is that Martin gave in his presentation a very good overview of making a decision and how it impacted the local innovation system in terms of who participates in it, the type of trades, for example, the types of products that they put out. And that excitement about innovation is not there for GMOs in South Africa. And while we have now this discussion, is these potential tools that reduce the barriers to innovation on the technical level, if it's not reduced on the commercial and the product level, it still leaves us exactly where we've been before. So we do need that certainty, like Lukesh now highlighted as well, to get the excitement, not only at the level of where Sanso operates and all the big innovators about cross-border movement, but also to get that excitement about genome editing innovation and the production of products that's relevant to South Africans, like Sinisizwe said in his presentation. In the end of the day, it's about the products that we can put in his case, the farmer's hands, but genome editing has the potential of putting products in a lot of other different people's hands. So with that, I would like to conclude from my side. It only leaves then the conclusion and I think the goodbyes from Chantel's side. I would like to thank all the presenters for their talks. Again, we will make this available, and everybody's got a contact detail on there. So again, it comes right back to the very first thing that I said today. It's not just about this webinar. Please use those talks to get the contact details of people, ask them these questions, engage. Biosafety South Africa has actually started a database for people interested in genome editing innovation in South Africa, where we can talk about things, where you can share things. In the end of the day, this is what it's about. If you're interested in participating in that, you can send me an email and I will put you on the list as well. So with that, thank you again for the presentations. I really enjoyed the presentations, and everybody that listened and participated in the discussion. It's much appreciated. Over to you. Thanks, Chantel. You off? Sorry, we still can't hear you.
Can't hear you, Chantal.
Yes. You on? No, it's not you.
That was me,
Lukesh.
Henny. I was saying that we couldn't hear her.
Yeah, oh, sorry. Thanks, Lukesh. Chantal, on my side, I can see you are muted. Ah, there it's off now. It's on again. Off. No, sorry, it's a technical issue. Sorry, I'm then going to take it into my hands. There's something issue at Chantal's side. So just again, from everybody's side, and now I must also thank Chantal and the USDA for putting this webinar together. And if we do not hear from Chantal just after this will probably be the last one, but I see she's putting her hand up. So hopefully, Chantal, we can hear you now. Yes. No, sorry, still not.
Henny, do you have access to the poll results?
Oh, yes, yeah. Thanks for reminding me about that. Can we please share that before we go? I don't know if I'm supposed to narrate this, but the first question about what is genome editing, and you've seen there, and the great majority of people say that it's all above. So it's about deletions, replacing, inserting DNA sequences or whatever. Thank you. Number 2. Maybe I've made an error. Oh, yeah, there you are. Thank you. Have you heard of the terms SDN1, 2, and 3 and understand the differences between them? Yes, 42 and no, 58. This is very important information to me. And this is exactly what we can do and follow up on webinars like that, is to make sure that when I've made a summary here of all the presentations, so we will ensure to include the detail on this there. It's not the time to explain it now, but it's a very important aspect to understand because it's the potential differentiation between, or your thresholds for regulation can be impacted by this. Next one, please. Do you think genome editing technologies could reduce the technical and cost barriers to bioinnovation, significantly increase the number of range of products and innovations? It's pretty clear. 94% of people, there's 1% that do not think so, and 2 people that do not know. We can continue to the next one. Adopting a fit-for-purpose and adaptive policy on genome editing is essential for advancing bioinnovation and improving the competitiveness of South African agriculture. It's almost the same outcome as the previous one, very important. And I think in context of what we're discussing today is innovation in the agricultural environment, these are very critical opinions, in my opinion. Next one, please. Increasing public dialogue and consultation on genome editing technologies is an important consideration for building societal trust and ensuring the acceptance of genome editing. 96% people, there's 2 that do not agree with this or didn't know, but I think again, this is one of the things that stood out. It's one of the questions that we've discussed repeatedly previous time and today. In the end of the day, innovation and what we refer to as sustainable innovation is a product that people want and that does not have impacts on the environment or on their health, obviously. So we do need to get buy-in and social license to do the innovation that we would like to do. So that communication, and I would like to put a plea out there to everybody that's involved in this field to please do your share. And you think it may not contribute, but in the end of the day, everybody's but that they do contribute to a better understanding. I think this is the last one. You can just confirm that for me, please. Yes, I think so. And with that, and Chantal, would you like to try just one more time? I see you are not muted. No, sorry, we still can't hear you. So then allow me for the final time just to, on behalf of Chantal, to thank everybody. And Chantal, like we said earlier, you were the center, the organization, or at the center of the organization of this webinar. We really do appreciate that.
Thank you.
We can hear you.
Oh, excellent. Wow.
Yes.
Sorry about that. It was just not my day for glitches. I think, thank you again to all the speakers. I think your contributions were very valuable. And I think we've all learned a lot, and that we are walking away better informed, on the prospects of genome editing and what it can do for agriculture. Of course, there's still a lot of, I think, key questions, and those were raised this afternoon. And so I think lots for us to chew on, as we contemplate what exactly South Africa's final outcome will be on the regulation of genome edited products. But also big thank you to Henny for officiating today's discussions and also keeping us on time. And also thank you to all of you as the audience who joined and participated. I think thanks also for being an interactive audience. And I know there was a question on the presentations. Both the previous event and this event has been recorded. So please do look out for those recordings on the CropLife South Africa website in the coming weeks. And that is it from me. So thank you, everybody, and have a good rest of your Tuesday. Thanks.
Thank you. Bye-bye.